Hello guys, this is Vineet, and there was a little concept that was left behind in the chapter properties of the DNA. That is, how do you purify the DNA using cesium chloride density gradient centrifugation? Okay, so what you have to do is you have to transfer the nuclear genome or DNA into cesium chloride solution and do the centrifugation at high speeds. Okay, around 4,50,000 xg for overnight, and then you have to transfer the entire solution to a gel membrane, agar gel membrane, where it gets separated. So that when you observe the gel membrane, the DNA settles in a band where the density is equal to the density of cesium ions. Okay, why it happens? Because the cesium ions bind to the DNA molecules and push them in the gel membrane. Okay, so the DNA which is settling at a band where the DNA density is equal to the cesium ions. Okay, so that point or that band is referred to as the neutral buoyancy band. Okay, so this happens in the normal DNA sequences which are equal in the adenine, guanine, cytosine, and thymine percentages. Okay, but if there is any variation in the guanine and cytosine percentages, you will get this two separate bands. Okay, one band having guanine and cytosine rich regions, so that is uh, higher density band around 1.701 gram per centimeter cube, and one band on the gel membrane at which indicates the 1.690 gram per centimeter cube. Okay, so the highest band which has the higher bond density or the density is also referred to as a guanine cytosine rich band, which is the main DNA band or the unique genome sequence band. Okay, which mainly involves the coding sequences. Okay, and the rest smaller band, or the smaller density band, this represents the repetitive DNA. Okay, and when you do the spectrophotometric studies, when you pass the 260 nanometer wavelength of light through the solution, okay, for quantifying the DNA, you will get two peaks when you plot a graph for this absorbent of the DNA against the buoyant density gradient. Okay, so main peak indicates the guanine and cytosine rich regions, and the lower peak will indicate the satellite peak. Okay, satellite DNA peak. Okay, even though we are getting two peaks. Okay, and the density is quite less. Okay, if you observe the density, there is only 0 0 0.1 gram per centimeter cube. Okay, so that 0.1 density is major variation between the main DNA and the repetitive DNA. Okay, so because of this less variation, you cannot separate the repetitive DNA and the guanine cytosine rich DNA or the unique DNA using this ultracentrifugation or density gradient method. Okay, so this is what the studies on this purifying the DNA tells about. Okay.